Hello everyone, my name is Grace Pan, and this summer I've been at NCSSM Durham as part of the M3 program working in the Matsunami lab at Duke University, which focuses on the study of olfactory receptors. My project topic is characterizing odor responses of chicken olfactory receptors. Until recently, there were many misconceptions about the importance of olfaction in birds. People believed that birds had a functionally insignificant sense of smell due to their anatomical structures and the observed behavioral responses. However, research has now shown that some avian species have even more olfactory receptors than the current highest count in mammals. This is important for future conservation efforts and to understand how we can use birds in studying olfaction. So, my research question is, what odors do chicken olfactory receptors respond to? Through the testing of olfactory receptors, we aim to enhance our understanding of avian chemosensation and genomic diversity by mapping out the reactions between our cloned receptors and diverse odors. To analyze olfactory receptors, we cloned our olfactory receptor from the chicken DNA and verified its presence through PCR. Then we attached a C-terminal tag that allowed us to easily identify the olfactory receptor, and we verified the tag through gel electrophoresis. As you can see in the screenshot attached above, the highlighted regions under the purple ones indicate that the tag was there. Now we have a complete plasmid with the olfactory receptor and the tag. In birds, 80% of their olfactory receptors are part of the subfamily called gamma C. However, gamma C has 6 different clades, so we selected 7 gamma C olfactory receptors from these different clades. As you can see on this chart, we cloned them out from 3 of them. So in the blue one, there's a 7 olfactory receptor, and then in the pink one, there's a 6, A4, and A8, and so on. Lastly, we inserted our plasmid with the tag and olfactory receptor into mammalian cells to run a glow sensor assay, and this allowed us to test our 7 cloned out olfactory receptors, 2 positive controls, and 1 negative control against 42 structurally diverse odorants. The glow sensor reagent that we used that we inserted into the cells contains CAMP, something that causes the cells to glow in the machine depending on how strong the reaction to the odor is. Here are the heat map results from Python. The brighter the color, the stronger an olfactory receptor responded to an odor. So if you look over here, based off of the key, the red regions are the positive controls and the negative control is boxed in orange. So the negative control is PCI, which is essentially a plasmid but without the olfactory receptor. So we expect it to have no response, which you can see from the purpley area. And then, meanwhile, the positive controls are previously tested olfactory receptors that we already have expected results for. So we expected them to either respond to a few specific odors or to respond to an abundance of odors. As you can see with WS8, it's responded to an abundance of odors. Now we will look at the olfactory receptor A1's response to just indole, which was very strong, as you can see by the bright yellow and matching it with the key. And that's the region that is boxed within the pink. So these figures display just A1's response to indole. My raw data, time normalized, PCI normalized, and both normalized are shown in these graphs. By normalizing PCI, our negative control, and time, we've removed background factors that could have possibly impacted our data. So for example, if one of the well plates had more cells than some of the others, that could have given us different data. Overall, we were able to successfully clone out 7 different chicken gamma C receptors. 2 out of the 7 responded strongly to a variety of odors, and that was A1 and A3. 1 out of the 7 responded specifically to beta alanine, and 4 out of the 7 didn't have a strong response to any of the odors. In the future, for our short-term goals, we plan to continue testing odors on the 4 non-responsive olfactory receptors to try to target which odors they might respond to. We also want to try cloning out gamma C olfactory receptors from a different clade because as of right now, we've only cloned them out from 3 clades. For more long-term goals, we want to consider the ecological and behavior relevance of these odors to chickens. And here are my references. And finally, I want to give a big thank you to the Matsunami Lab, especially Hiro Matsunami, Wendy Sun, and Robert Driver for helping me and teaching me throughout this process, and the NCSM Foundation and Burroughs Wellcome Fund. Thank you!